Now let's talk about the next generation cell culture technology. So what are the technologies that have been developed in the field of cell culture? So in cell culture, we have 2D monolayer and versus the 3D spheroid. So it means here the cells are grown in just a 2D through two-dimensional, and then cells can also be grown in a three-dimensional shape. Right, 3D cell culture, cells are grown as cell spheroids or cell aggregates to form a three-dimensional structure of the original tissue. So instead of having a flat layer of the cells, we can actually grow the cells into a specific shape that we want. Since they highly resemble the original tissue, the experimental outcomes of a 3D cell culture can be easily extrapolated with that of an in vivo response. The additional dimensionality of 3D cultures influences the spatial organization of cell surface receptors. The 3D spatial arrangement affects a wide range of cellular functions like cell proliferation and differentiation, cell signaling, gene expression, etc. So these are the comparisons between 2D and 3D cell culture. What's the difference between these two techniques? So 2D cell culture, plastic is used as substrate. So the petri dish or the flask is uses plastic as the ingredient or component. And 2D cell culture is unable to mimic in vivo microenvironment and lack cell-to-cell or cell-to-matrix interaction. And cells in a 2D culture might lose their morphology. The morphology or the shape of the cell may be different to the primary cells. But it is economical because it's very easy to do a 2D cell culture and it does not require additional apparatus to run 2D cell culture. And it is very useful for high throughput capacity, feasible for mass screening of drugs. For example, you want to test thousands of drugs. You don't want to use 3D cell culture to see the effect of the drug on the cells because it's going to cost a lot and it is also time consuming. Instead, you can just use 2D cell culture to expedite the process and to lower down the cost. And for 3D cell culture, the natural extracellular matrix is used as the substrate, mimic the in vivo microenvironment as well as cell-to-cell and cell-to-matrix interactions. Physiologically, 3D culture highly resembles its source tissue. However, it is expensive and laborious and relatively low throughput capacity, but you must use this 3D cell culture in the field of tissue engineering and regenerative medicine. So how to do the 3D cell culture? There are a few techniques to do the 3D cell culture, namely scaffold-based, non-scaffold-based bioreactors and gels. So just imagine you want to build a house. In order to build a whole house, of course, you need the foundation, and then you need the beam, and then you need the pillars, and then you need a few frames. Then you can build the wall. You can put the bricks to build a complete house, and then you can put the roof on top. So similar thing with 3D cell culture, you need to have a base in order for the cells to grow. So this base is called the scaffold. So the scaffold base can come from polymer, and then second one is biologic, and then the third one is micro-patterned surface microplates. And then for non-scaffold base, there are hanging drop microplates, spheroid microplates containing ultra-low attachment coating, microfluidic 3D cell culture, and then next we have bioreactors and gels. So first, for polymeric hard scaffold, typical size is 150 to 200 micrometer, and lactite and glycolide copolymer may be used as materials. So these are the polymers, lactite and glycolide. Uh, in regenerative medicine, cells are grown on this scaffold for transplantation purposes to replace defect tissue, widely used for engineering bone, cartilage, ligament, skin, vascular, neural, and skeletal muscle tissues. In preclinical in vitro testing, cells are grown to model tumors in the lab. So these polymeric heart scaffold are normally used to grow tissues for transplant. But for organ transplant, polymeric heart scaffold is not suitable because it can cause side effect in human body. It can cause immune response in the body because of the foreign material of the polymeric heart scaffold. Therefore, biological scaffolds can be used. So biological scaffolds are produced from natural components of biological origin, for example, proteins such as fibronectin, collagen, laminin, and gelatin. Cells attach and reorganize into desired organs 3D structure. So if you can see in this picture, the biological scaffold here is the ear, and then the researcher pipette out the cells onto the scaffold, and then the cells will grow covering the scaffold of the ear. So these biological scaffolds provide suitable microenvironment for the anatomical and physiological development of cells. They are widely used in regenerative medicine as well as tissue engineering. And this is the third type of scaffold base, which is the micro-patterned surface microplates. Each plate contains micrometer-sized compartments arrayed on the bottom, usually coated to create a low adhesion surface. You don't want the cells to adhere to the bottom of the plate too much because it's going to be hard to detach the cells from the bottom, and that's why it's coated. The bottom is thin and transparent for microscopic imaging, and they are usually used in drug testing, cell biology analysis at the molecular level.